we begin, I want to acknowledge that we live, work, play, and participate in a community on the unceded, ceded, and traditional territories of the 203 First Nations, along with 38 Métis chartered communities, each of which possess their own unique traditions and history here on this land that we now refer to as British Columbia. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action, the United Nations Declaration on the Rights of Indigenous Peoples, and the BC Declaration on the Rights of Indigenous Peoples Act. In all of our work, we're committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I'm joining you from Vancouver, which is part of the unceded homelands of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. And I recognize that many of you are not calling in from even Canada or Vancouver, so I encourage you all to do some research on the Indigenous cultures in your community. I'll just very briefly tell you a little bit about the Women's Health Cluster. We have a number of members, over 350 actually, worldwide across 17 different countries. And we run a number of events, such as the Women's Health Seminar Series, which today is the last one from the 2021-2022 session. You can get a certificate if you've been attending about 90% of them, so please reach out and get your certificate if you'd like to do so. We're going to have a Women's Brain Conference in 2022 in October, November on how hormonal contraceptives affect the brain. And I'm sure there'll be some more trainee events coming up. We just ran a competition for travel awards. I don't know when those will be coming up, but pretty soon. So you'll hear about them soon, but please do join the cluster so you're eligible for those awards and become a member. Donate if you feel so inclined, and we are very thankful to our current sponsors, including UBC. So now I'm going to stop sharing my screen so that I can introduce Dr. Barha while she starts sharing her screen. And Dr. Cindy Barha is a postdoctoral fellow in the Department of Physical Therapy at UBC, affiliate member of the Women's Health Research Institute, and an adjunct professor at Simon Fraser University. Dr. Barha is a translational neuropsych scientist with training in behavioral neuroscience, cognitive neuroscience, evolutionary biology, epidemiology, and neuroendocrinology. Her research examines how genetics and hormone-related life events interact to alter cognitive and brain trajectories across a lifespan, and to determine how these factors affect pharmacological and non-pharmacological treatment efficacy in older age. Currently, Dr. Barha is examining sex differences in the effects of exercise on brain health in older adults. She's currently funded by the Alzheimer's Association and Brain Canada. And her title of her talk, you can see there, is Walking for Brain Health, the Role of Biological Sex and Reproductive Experience on Exercise Efficacy. I know Dr. Barha very well. She, can I say, started out her grad career in my lab. I could have come up with a bunch of fun facts, but she gave us one, so I'll read out hers. And I think many of us maybe can relate to this fun fact. Dr. Barha is obsessed with the Great British and Canadian baking shows. She's currently watching the Great Australian Bake Off. I have not watched that yet. I am blaming those shows for my 10-pound COVID weight gain, by the way. Her goal is to get her father-in-law onto the Great Canadian Baking Show, so I'm really excited to hear about that. He's a phenomenal baker, and her 3-year-old son always calls him Grandpa Cakes, and they even bake together. And Cindy says she's not a good baker, but that doesn't stop her from trying. So without further ado, I welcome Dr. Cindy Barha. Thank you so much.
You can see my slide. Everything's good? All right. Thank you, Lisa, for that wonderful introduction. When you asked me for my fun fact, at first I was like, there's nothing fun about me. I'm just boring. But I was watching the Great Australian Bake Off at the time. And so I was like, oh, this is it, because I am absolutely obsessed with that show. And the Australian one is even better because the hosts are hilarious. So I definitely think you need to watch it. So first of all, I just want to thank everyone for taking time to be here on Zoom today. It's an absolute honor to be invited to present at this Women's Health Seminar Series and to be actually the last speaker of 2022. I don't know about you guys, but I can't believe it's April already. I really think the pandemic has sped up time, to be honest. Okay, so let's get started. So I'm sure we're all aware that dementia is a global health issue. So worldwide, 1 new case of dementia is diagnosed every 4 seconds. And by the year 2050, it's projected that over 131 million people will suffer from dementia. So 2/3 of all people with Alzheimer's disease are female, and females have a greater lifetime risk of Alzheimer's disease, which is partly related to their 4.5-year longer lifespan that they have. Females also show greater cognitive decline after being diagnosed, and in both humans and in rodent models, females exhibit greater Alzheimer's disease neuropathology, and this includes faster hippocampal atrophy and greater amyloid beta plaque deposition. As well, some Alzheimer's disease non-modifiable risk factors show a sex difference and have a greater impact in one sex. So for example, the ApoE4 gene variant, which is the greatest genetic risk factor for Alzheimer's, has a stronger effect in females than in males. Other risk factors are actually specific to one sex. So for example, pregnancy, oophorectomy, and natural menopause are all sex-specific risk factors found in females. So in addition to non-modifiable risk factors, which do account for about 60% of dementias worldwide, there's been 12 modifiable risk factors that have been identified that account for about 40% of cases. So these factors include things like smoking, alcohol use, head injury, and diabetes. And I've shown the 12 here on the slides. So elimination or even reduction in any one of these risk factors will have a substantial impact on dementia prevalence. Thus, developing interventions that actually target one of these modifiable risk factors is, of course, of utmost importance. So like I mentioned before, while some risk factors are unique to one sex, like menopause in females, others may be actually relevant to both sexes, but may still be more prevalent or stronger in one sex. So importantly for my work, physical inactivity is one of these modifiable risk factors that may actually show a sex difference. It may be even more important in one sex. So some recent work published last year out of Finland using combined data from 3 Nordic population-based cohort studies for a total of about 2,300 people suggests that physical inactivity is a significant risk factor for dementia in females, but maybe not in males. So in this table from the study, you can see that in females, which I've marked down here in red, physical activity is significantly associated with a reduced risk of dementia, as shown by a hazards ratio of less than 1. In males, which I've shown here in blue, the association between physical activity and risk of a dementia was actually not significant. And that significant interaction in that last column there in green between sex and physical activity shows that the association between physical activity and dementia risk was only significant in the females, and that significant level was different between males and females. Thus, this data supports this idea that physical inactivity is actually a stronger risk factor among females, and that increasing physical activity levels may be actually more important for dementia risk in females than in males. Importantly, no effective pharmaceutical approach is currently available for dementia or for age-associated cognitive decline. However, lifestyle factors, which includes physical exercise, actually show greater promise in both the prevention and treatment of cognitive impairment. So engaging in exercise does seem to be beneficial for cognitive function. And you can see that in this seminal review paper by Stanley Kokomi and Art Kramer, which really kind of set this whole field of exercise neuroscience in motion. So exercise, which is in the white bars, is associated with benefits to several different cognitive domains versus control, which is the gray bars. And in this particular analysis, exercise had its greatest effects for the domain of executive functions. Okay, so before I go on, I really just want to make sure everyone is on the same page, as I will be talking about executive functions quite a lot in today's talk. So executive functions are the set of mental processes that allow us to do things like plan, focus our attention, remember instructions, and really juggle multiple tasks in a successful way. So really, your brains need this set of skills to filter distractions, prioritize tasks, set and achieve goals, and control impulses. So in this particular review paper, exercise interventions had their greatest beneficial effects on tests that tapped into these different cognitive processes and skills that are under that whole umbrella term of executive functions. But despite these promising results, a lot of variation still actually exists in exercise efficacy on cognition, as not all studies find this beneficial effect. So for example, the LIFE trial, which is one of the largest and longest exercise randomized control trials that's ever been conducted with over 1,600 older, cognitively healthy people, found that a 2-year multimodal exercise intervention actually didn't increase performance across several different domains, including hippocampal dependent learning and memory tasks, or even executive function tasks. And this was compared to a health education control group. As well, this 2-year multimodal exercise intervention actually didn't even increase or decrease the incidence of dementia. Thus, a major goal of exercise neuroscience really as a field is to enable precise evidence-based recommendations for brain health by better understanding the sources of this variation in study outcomes. So to do this, we examine the what, the who, the how, and the when of exercise. So what type of exercise is best for cognitive health? So is aerobic exercise better? Is resistance training better? Is there a better combination of the two? Or are alternative forms of exercise like yoga and Tai Chi or even dance more beneficial for cognition? For whom each type of exercise is most effective in promoting cognition, this includes looking at things like sex differences, looking at specific genotypes, as well as looking at the cognitive status of individuals, such as individuals that might be at more risk for dementia. We also examine how different forms of exercise are exerting their influence on the brain, including looking at things like neuroimaging outcomes, looking at neuroplasticity markers, neurotrophic factors, hormones like estradiol, testosterone, and cortisol, and even looking at things like sleep and the immune system. And finally, we also examine when exercise is most effective at bolstering brain health by taking more of a sort of lifespan approach. And so, for example, looking at midlife versus older age. And so for my part, I've mainly focused on examining the who and the how by first asking, do sex differences exist in the effect of exercise on cognition? But I do want to point out that in the near future, I am hoping to look more at the when of exercise and to see if it's influencing cognitive health at different parts of the lifespan. So to address this goal of enabling precise evidence-based exercise recommendations for brain health, I employ a multidisciplinary translational approach to my research. So I followed for part of my sort of earlier career, the more typical paradigm of going bench to bedside. So taking what I learned in the lab and applying it in a clinical setting. However, more recently, and as I hope to transition to a faculty position at some point, I do endeavor to go more from bedside to bench. So utilizing appropriate rodent models to sort of further delve into the underlying mechanisms of those effects that I'm first seeing in my human studies. So this is in essence flipping that typical paradigm, as it does offer some unique advantages to explore potential mechanisms that really aren't quite tractable in the clinical setting. And so I'll give an example near the end of my talk as to how I've been able to apply this research approach to myself. So ultimately, I really do believe that this sort of research approach will allow for the translation of discoveries into clinical guidelines, as well as new developments. And I really do believe that this could help change clinical practice. So as a first step into looking at potential sex differences and looking at the sources of variation, I looked at potential sex differences in exercise effects for cognition. So I conducted a meta-analysis of 39 randomized control trials that were done in older, cognitively unimpaired adults. So we grouped these studies into those that used aerobic exercise, or those that used only resistance training, or the studies that used sort of a combination of the two. To start looking at sex differences, the studies were then split into those with a high percentage of female participants and those with a lower percentage of female participants. We weren't able to merely look at male versus female only studies, as all of the studies did include participants of both sexes. So we also looked at several different cognitive domains, but I'm going to focus on executive functions for today's talk, as that was the domain that showed the greatest effect sizes. So for studies of aerobic training, we found that overall the effect benefited aerobic training compared to controls. And so that's the black diamond on the bottom of the graph. And we found that the effect of aerobic exercise on executive function was actually greater in studies with a high percentage of females, which is the purple diamond, compared to studies with a lower percentage of females, which is the blue diamond coming up. For resistance training, we found that overall, the effect did benefit the resistance training group compared to the control, so that's the black diamond. And similar to what we found for aerobic training, there was a greater effect of resistance training in studies with a high percentage of females, which is the purple diamond, compared to the low percentage of female participant studies, which is the blue. And then finally, for multimodal training, so that's when resistance training and aerobic training were used together, we find the same thing. The greatest effect was in the high female studies compared to the low female studies, so the purple versus the blue. Importantly, my meta-analysis was actually included in the World Health Organization's guidelines as evidence for exercise as a strategy for reducing dementia and cognitive decline. And I think this is kind of a nice example of how this research approach can actually lead to translation of discoveries into guidelines. I also have another meta-analysis looking at aerobic training studies with cognitively healthy older rodents. So these are older rats and older mice. And we were able to look at voluntary aerobic training paradigms, which use running wheels, separately from studies that used forced aerobic training paradigms that typically use treadmills. And we found that voluntary aerobic training benefited spatial learning and memory, so that's the black diamond, compared to controls. And the effect sizes were actually similar between studies that were done only in females, the purple diamond, and studies done only in males, so the blue diamond. Forced aerobic training also benefited spatial learning memory, so that's the black diamond. But notably this time, the effect size was actually greater in female-only studies, so that's the purple diamond, compared to male-only studies in blue. So to begin to look at how aerobic training could be influencing cognition differently in males and females, I looked at the effects of aerobic training on brain-derived neurotrophic factor, or BDNF, in this rodent meta-analysis. So BDNF is involved in neuroplasticity and neuronal health, and you can basically think of it as a vitamin for your brain. And previous work has actually shown in both rodents and in some human studies that BDNF might be mediating the effect of aerobic training on the brain. So as we expected, we found that the overall effect size benefited the exercise group, which is the black diamond. So aerobic training significantly increased BDNF levels compared to controls. The greatest effect of aerobic training on BDNF was actually seen in studies with females only, which is the purple diamond, compared to male-only studies. So you can see this is quite a large effect size difference. So to me, this suggests that, that BDNF may be one possible mechanism that is underlying that sex difference in exercise effects on cognition that I've been seeing. I've also been able to more sort of directly examine the interaction between biological sex and aerobic training by conducting a sex differences analysis of a completed randomized control trial by our lab. So this was a 6-month randomized control trial with older adults with mild vascular cognitive impairment, which is the second most common form of dementia after Alzheimer's. So half the participants received this 3 times per week, moderate paced walking program, and the other half were in the control group and received usual care and educational material. So the findings of that primary paper did show that aerobic training significantly improved episodic memory performance and also significantly reduced blood pressure compared to the control group at trial completion. But for my sex differences analysis, I actually focused on executive functions, as that was the cognitive domain that my meta-analysis had pointed towards as having the largest effect sizes. So this graph here is showing change in performance on the trail making test, which measures set shifting, a component of executive functions. So here, negative change scores actually indicate an improvement in performance from baseline. And you can see that in the females, the aerobic training group in dark purple significantly improved executive functions on this test compared to controls in the light purple. In males, the aerobic training group actually didn't significantly differ from the control group. And if you compare just the 2 aerobic training groups, those 2 dark bars, you can clearly see that females did much better after exercise on this particular executive function test compared to the males. Okay, so at this point, I usually just leave the story at females show greater cognitive benefits from exercise. But I'm sorry, guys, you're not off the hook, of course. Exercise is, of course, still good for men. So for example, although we didn't see the benefit to executive functions from aerobic training for males, we did see these fantastic improvements in cardiovascular function, so functional fitness capacity, as measured by the 6-minute walk test. And we didn't see this improvement in females. So I find this particularly intriguing, considering this hypothesis that aerobic training is actually improving brain health via improvements in cardiovascular fitness. So if in females, it's not cardiovascular fitness that seems to be mediating the effect of exercise on the brain, what could it possibly be? Well, I turn once again to BDNF, my favorite thing. That's that growth factor involved in neuroplasticity and neuronal health, that brain vitamin. And I found that at trial completion, aerobic training significantly increased levels of BDNF to a greater extent in females than in males. And so this matches what I found in that rodent meta-analysis that also found greater effects of aerobic training on BDNF levels in the female rodents. Okay, I've also been able to look at this potential sex difference in exercise efficacy in data from a 10-year longitudinal epidemiological cohort study called the Health, Aging, and Body Composition Study, or Health ABC. So in this study, about 2,900 older adults were recruited from Memphis and from Pittsburgh in the States. And at intake, the participants were between 70 and 79 years of age. Though not a major component of the study, the researchers did assess physical activity levels through self-reported time spent walking. So this is minutes per week. And this was measured annually from years 1 to 10 of the study using a standard questionnaire. So we were able to actually look at individual trajectories of change in time spent walking. And on average, in this cohort, time spent walking actually decreased by about 8.4% every year. So we fit linear mixed models to the data to calculate each individual's initial time spent walking, so that would be the intercept of the model, as well as each individual's change in time spent walking over the 10 years. So that's the slope of the model. So here, a negative slope indicates declines in the amount of time spent walking. Although cognitive functioning was also not a main component of this study, executive functioning was assessed with a digit symbol substitution test, which measures processing speed, sustained attention, and working memory. And global cognition was measured with a modified mini mental state exam, or the 3MS. So both of those tests were given several times over the 10-year study period. And so we were able to look at individual trajectories of change in performance on each of those tests over that 10-year period. So once again, we fit linear mixed models to the data for each test separately to calculate each individual's initial performance, so the intercept, as well as each individual's change over time in performance, which would be the slope. So using latent growth curve modeling, we actually estimated 4 paths of interest for each of our 2 cognitive outcomes, so the DSST and the 3MS. So, okay, let's walk through this. Path A is looking at the association between initial time spent walking and initial cognitive performance. Path B is looking at the association between initial time spent walking and linear changes in cognition, so the slope. Path C is looking at the association between initial cognitive performance and linear changes in time spent walking. It's a mouthful. And then path D is looking at the association between linear changes in time spent walking and linear changes in cognition. So these models were stratified by sex, so we could determine whether any of those 4 paths actually varied between males and females. And separate models were run for DSST and for the 3MS. And we adjusted each model for baseline age, education, clinical site, so whether it's Pittsburgh or Memphis, race, BMI, prevalent diabetes, coronary heart disease, and cerebral vascular disease. So here first are the results for the DSST. So that's what measures processing speed and executive functioning. So the numbers down here are standardized betas. So they tell us the correlation or the strength of the relationship between the variables that we're looking at. And red betas are for females and the blues are for the males. And you can see that both path B and path D were significant, but only in females. So path B indicates that higher initial time spent walking was associated with a less decline in executive function over time. Path D indicates that maintenance or even maybe increases in time spent walking over the 10 years was associated with less decline in executive function through the 10 years. And these were only seen in females, not in males. Next for the 3MS, so this is looking at global cognitive function, we can see that the path D was actually significant, but this time in both males and females. So this indicates that in both sexes, maintenance or increases in time spent walking was associated with less decline in global cognition. Okay, so to examine whether this relationship between physical activity and biological sex can be reflected within the brain, I use data from the Healthy Brain Project, which is a sub-study of the Health ABC project, in which participants underwent neuroimaging at year 10. So I utilized the data to explore the relationship between time spent walking and the volume of the dorsolateral prefrontal cortex in both males and in females. So this brain region was selected a priori, considering the really strong evidence that it shows age-associated decline and deterioration. And it's also the area that subserves executive functions, like what is being measured with that test called the digit symbol substitution test. So after controlling for race, age, education, BMI, diabetes, cardiovascular disease, cerebral vascular disease, brain atrophy, and total gray matter, we found that in females only, greater increases in physical activity in the form of walking over the 10 years was actually associated with a larger volume of the left dorsolateral prefrontal cortex, which is similar to what we found for that relationship between walking and performance on the DSST, that executive function test. So I provided converging evidence from 3 types of studies: meta-analysis, randomized control trials, and epidemiological cohort study, that really suggests that a sex difference exists in aerobic exercise efficacy for executive functions, with females actually showing greater cognitive gains. So this may be related to aerobic trainings having a greater effect on prefrontal cortex volume and BDNF levels in the females. So in addition to sex differences, I'm also interested in looking at how the cognitive status of individuals can actually moderate the efficacy of exercise to improve brain health. So specifically, I was wondering if the effectiveness of exercise in females could be dependent on previous reproductive history. So this is because emerging evidence suggests that previous parity can alter a female's risk for dementia later in her life. And so we've heard a little bit about this in previous talks in the seminar series. So we know pregnancy has profound effects on female physiology, brain function, and disease risk in both the short and the long term. So I'm not going to go into this figure in too much detail, but very briefly, during pregnancy, there are large adaptations across several different systems, including decreases in total brain volume, hippocampal volume, and memory function. During pregnancy, immunoregulation improves and steroid hormone levels increase 200 to 400 times normal levels. And it's believed that these changes in immunity and hormone levels may actually lead to some of these sort of long-term consequences that pregnancy has for female health. In the short term, in the brain, there are reductions in gray matter in several different brain regions, including the hippocampus, as well as declines in neurogenesis and increases in cognition. However, I do want to point out that these brain and cognitive changes do seem to be dependent on the number of previous pregnancies. So, for example, first-time pregnancies versus second and third-time pregnancies have different effects on cognition and neuroplasticity in rodents. So much less is actually known about the long-term effects of previous parity in middle and older age. So parity does seem to increase susceptibility to things like obesity and metabolic disease in older age, but actually seems to decrease risk for several different cancers. Even less is known about the long-term effects on the brain and on cognition. However, some studies do find that the risk for Alzheimer's disease seems to increase with high parity, with 5 or more pregnancies associated with increased risk. As well, nulliparity, so having 0 pregnancies, has also been associated with a greater risk of Alzheimer's disease. So to see whether previous parity can alter this relationship between physical activity and cognitive aging, I once again utilized data from the Health ABC study. So as I described before, the researchers did assess cognition longitudinally over 10 years. So global cognition was measured with the 3MS, and executive functioning and processing speed was assessed with a digit symbol substitution test. And I was able to look at average cognitive performance and change in performance over time. The researchers also assessed physical activity levels through self-reported time spent walking, so minutes per week, that was measured annually from years 1 to 10. And for the analysis that I'm going to present now, I actually looked at average physical activity over that 10 years using this measure. I broke down total parity, so the total number of complete pregnancies, into 4 groups of females based on previous literature. So we have a nulliparous group, so these are females with 0 complete pregnancies. We have a low parity group, so these are females with 1 or 2 complete pregnancies. We have a medium parity group, so these are females with 3 or 4 complete pregnancies. And finally, we have this grand multi-parity group. So these are females with 5 or more complete pregnancies. To examine the interaction between average physical activity and the parity group, we ran ANCOVAs with average cognition and change in cognition over the 10 years as our 2 dependent variables. When there was a significant interaction between physical activity and parity, we extracted simple slopes to look at the association between average physical activity and the cognitive variable that we're looking at for each parity group separately. And in our analyses, we controlled for a whole slew of health-related covariates, including things like depression, education, race, age, diabetes, ApoE4 status, smoking, alcohol use, cardiovascular and cerebrovascular disease, hypertension, blood pressure, and family income. Okay, so on the y-axis on the next few graphs, we're going to be looking at the association between average physical activity and the cognitive variable being examined. So for example, in this graph here, each of these bars depicts the strength of the association or the correlation between average physical activity and average score on the 3MS for each parity group. So specifically, we see that the association is significant in the nulliparous group and the grand multi-parity group, so the first and last bars. indicating that in those two groups, more average time spent walking was associated with better average global cognition. And in the low and medium parity groups, so those are our two middle bars, average global cognition was actually not related to physical activity. So to examine whether previous parity could be contributed to that sex difference in exercise-induced cognitive gains that I've been seeing that favors females, we also looked at this association in males and compared it with our 4 parity groups. And we actually found that the association between average physical activity and global cognition was not significant in the males. And we see the same thing with average executive functioning. The nulliparous group and the grand multi-parity group, so the first and last bars, showed better executive functioning with more physical activity. And once again, the association was not significant in the males. For change in global cognition over time, we found that the association between average physical activity and change in 3MS over the 10 years was significant this time in only the grand multi-parity group, so that last bar, indicating that in this group, more time spent walking was associated with less decline in global cognition over time. And once again, the association was not significant in the males. And we see the same thing for change in executive function over time. The grand multi-parity group showed less declines with higher average physical activity, and the association was not significant in the males. Thus, it seems that the greatest cognitive benefits of physical activity may actually be seen in the most at-risk groups. So the most at risk for cognitive decline and potentially dementia. So specifically, we saw a stronger association between physical activity and cognition in females with the highest parity, which is also possibly the group that's most at risk for Alzheimer's disease, as been previously shown. As well, we saw a strong association between physical activity and cognition in the nulliparous females. So nulliparous rats, shown here in the light blue in this graph, have been shown previously to have reduced working memory compared to the multiparous females, which are in dark blue. And we've also shown that nulliparous females have reduced reference memory compared to the multiparous group. And also the nulliparous group in light blue have lower levels of hippocampal neurogenesis in older age compared to the multiparous females in the dark blue. Okay, so the final piece of this WHO puzzle, this part here, for exercise efficacy on cognition that I want to discuss today is genotype. So polymorphisms are when a gene comes in different forms. So there are several genetic polymorphisms that have been proposed to be drivers of cognitive aging. I'm actually interested in a common polymorphism in the gene that codes for BDNF, which, as a reminder, is that growth factor that's important for neuronal health and neuroplasticity. And it's also the molecule we've shown to be involved in how aerobic training may be affecting cognition, more so in females than in males. So the Val66Met polymorphism within the BDNF gene is present in about 20 to 50% of the human population, depending on ethnicity. So carriers of the polymorphism, so these are people with the Met allele, have altered intracellular trafficking of the mature form of BDNF compared to Val carriers. As well, some studies in humans find that the Met allele is associated with a smaller hippocampal volume and impaired cognition compared to Val carriers. But of course, not all studies are finding this. So one reason for these discrepant findings that is of interest to me is, of course, potential sex differences in this relationship between BDNF and the brain. So for example, estradiol modulates BDNF levels in both humans and rodents, with increases in estradiol leading to increases in BDNF. As well in humans, females show higher BDNF content in certain brain regions compared to males, and that includes the prefrontal cortex, which subserves the cognitive domain of executive functions. And we've actually shown a significant interaction between the BDNF Val66Met polymorphism and biological sex for cognitive decline in older adult humans. So we wondered whether biological sex was interacting with this BDNF polymorphism to moderate aerobic training effects on cognition. So to start to look at this in humans, I've conducted a secondary analysis of a completed randomized control trial of 6 months of aerobic training versus active control in older adults, in which I was able to look at the interactive effect of biological sex, BDNF genotype, so whether they're Val carriers or Met carriers, and experimental group, so aerobic training versus control, and the effects on executive functions using 3 different tests. So we found a significant 3-way interaction between biological sex, BDNF genotype, and experimental group for 2 of the 3 tests of executive functions that we looked at. So first, the results for the trail making test, which measures set shifting, a component of executive functions. So here, higher numbers actually mean worse performance because it took them longer to complete the task. And aerobic training is in gray and our control is in black. And we found that aerobic training actually improved performance versus controls, but only in the female BDNF VAL-VAL carriers. And aerobic training actually decreased performance in male VAL-VAL carriers. And the aerobic training and the control groups actually didn't differ in males or females that had the MET allele. So the other test we found a significant 3-way interaction for was a digit symbol substitution test, which measures sustained attention, working memory, and processing speed. So this time, higher numbers actually mean better performance. And we found that aerobic training significantly improved performance versus control, once again, in only the female BDNF VAL carriers. And this time, aerobic training actually decreased performance in the female MET carriers. And in males, aerobic training and control didn't actually differ regardless of genotype. Okay, to start looking at how aerobic training's effects may be altered by biological sex and the BDNF Val66 MET polymorphism, I'm looking at the role of neuroplasticity using the approach of going bedside to bench. So I'm currently working with Dr. Lisa Glia and 3 of her very wonderful trainees. So Dania Islis, Muna Ibrahim, and Talon Splinter to look at exercise effects on cognition and hippocampal neurogenesis using the BDNF Val66 MET mouse model. So before I go get into that study, I'm going to do a very quick reminder of hippocampal neurogenesis. So hippocampal neurogenesis is a production of new neurons that occurs throughout life and has been shown to occur in 1 million species that have been studied, including us and humans. So we can visualize these newly generated neurons in the dentate gyrus using immunohistochemistry procedures that stain cells expressing exogenous markers or endogenous neuronal markers, such as double cortin, which is specifically expressed in immature neurons up to 3 weeks of age, as shown in this lovely picture. So progenitor cells residing in the subgranular zone of this dentate gyrus can be induced to divide, producing daughter cells. At least one of those daughter cells will begin to mature, and within about 4 to 10 days, it will begin to extend an axon towards the CA3 region. If this new cell continues to survive, it will develop into an immature neuron by about 3 weeks of age, with short dendritic processes and a longer axon. And we know, at least in the male mouse brain, that this neuron will reach maturity in about 4 months. So when I speak about immature neurons, I'm going to be referring to all these stages of cell development. Okay, so back to that actual study that I was talking about. Okay, so we used older male and female mice that had the human BDNF Val66 MET polymorphism inserted into their DNA. So the mice were either MET carriers or VAL carriers. Half the mice were given free access to running wheels in their cages, and the other half were controls and were given access to locked running wheels in their cages. We trained and tested the mice on an associative learning task and an executive function task using the Bussy-Succida automated touchscreen chambers. The mice were then sacrificed at 16 months of age to look at hippocampal neurogenesis. So first, for the associative learning test in the MET carriers, so those are the rodents with that polymorphism in them, we found that none of the 4 groups actually reached the criterion of getting 70% of the trials correct in a given session, indicating that the MET carriers were not successfully learning this associative task. In the VAL carriers, the male exercise and control group did not differ, but the female exercise group did actually reach criterion and learn better than the female control group. For executive functioning in the MET carriers, although the female exercise group did learn a little bit better than the female control group, which learned absolutely nothing, none of the MET carriers actually reached criterion, regardless of exercise. And in the VAL carriers, the female exercise group reached criterion and learned better than the female control group, and the 2 male groups did not differ. So Talon Splinter, who I think is on today on the Zoom, has actually started looking at hippocampal neurogenesis in these rodents and found that when looking at the density of double cortin cells, so this is the density of immature neurons, aerobic training only increased the density compared to controls in the female BDNF VAL mice, but not the female BDNF MET mice. In males, aerobic training did not influence neurogenesis in either the VAL mice or the MET mice. So these neurogenesis results actually fit kind of nicely with our cognitive results, as it was the female BDNF VAL mice that also showed those cognitive benefits from exercise. And I want to also point out these results in mice actually mirror what we found in humans with the female BDNF val-val carriers showing that cognitive benefit from that 6 month of aerobic training intervention. So in conclusion, I've shown that in order to enable precise evidence-based exercise recommendations for brain health, we must take into consideration who the population we're working with is and how exercise is exerting its effect on the brain. So specifically, I showed that females seem to gain greater cognitive benefits from aerobic training, and this may be related to greater, sort of a stronger or greater relationship between exercise and the volume of the dorsolateral prefrontal cortex that's found in females, as well as it could be related to the aerobic training induced increases in BDNF that we see in females. As well, I've shown that previous parity is an important moderator to consider for the who part, as that is a factor that may moderate exercise efficacy. And it seems that the groups that are most at risk for dementia and possibly cognitive decline may actually be benefiting the most from exercise, as the grand multi-parity group and the nulliparity group show the strongest relationship between physical activity and cognition. And finally, I've shown that genotype matters, as aerobic training effects are greater in female BDNF val-val carriers, as seen in both our human study and our rodent study. I would like to thank you to my supervisor, Dr. Teresa Lou Ambrose, and my co-supervisor, Dr. Kirk Erickson, as well as all the trainees and staff that have helped, as well as thank you to my collaborator, and that includes my 2 main collaborators these days, my 3-year-old and my 11-month-old, which are not thankfully at home right now. And I'll come back to this funny cartoon, but I also want to do a shameless plug for a study that we're conducting right now. We're seeking 14 pregnant people between the ages of 18 and 40 who've recently, so less than 12 weeks, become pregnant with their first child to take part in the study. So this is a really fun study looking at memory and brain function. It's very COVID safe. So if anyone's interested, or if anyone knows anyone that might be interested, please take down our contact information.
Thank you so much, Cindy. I'm not currently pregnant, so I can't participate.
Darn.
Fantastic talk for those of you that are online. If you feel like unmuting yourself or doing the emojis, that would be great. To give a little clap to Dr. Barha. We actually have a question in the chat, which I actually think is a very good question from Cheryl. Cheryl, do you want to ask the question out loud, or do you want me to ask it for you? Oh.
No, I can ask it. I'm just wondering. I think the studies on the BDNF were really interesting, but I was also wondering whether anybody on the, that was part of those studies mentioned whether they were walking alone or walking with friends, because I know for myself, walking alone and walking with friends are 2 very different experiences, and just having that social interaction may actually be, part of the differences that you see. I'm thinking like, when you were talking about your, your multi-parity and, all that things like, people who have 5 children would benefit a lot by walking with another adult just for, 1 hour a week.
What are you talking about?
So I'm just wondering if that actually plays a big part or not. It's harder to, I guess, study in mice, but yeah, I'm just wondering if any of that was looked at.
So you're absolutely right. Social interactions are a huge, have a huge influence, even in these exercise studies. So the way we, I guess, what I think for the sex difference and even the parity one, not so much yet, because we haven't got to that part, but with the sex differences results, what's interesting is that we see them across different types of studies. So in the epidemiological one, so that's the one where we're looking at just time spent walking, we weren't able to, I don't think the researchers ever thought about looking at how the physical activity was being done. In fact, they used a really crude measurement, like time spent walking. It's not a good measure of physical activity. There's definitely better ones out there, but they didn't collect any information about social interactions during the walking. But what's interesting is that we still see that same sex difference when we look at randomized control trials. And we can actually look at the social interaction component by using a control group that would equate the exercise group and the control group on social interactions. And so the control group typically that we use would be coming into the lab and would be an active control group. So they'd actually be taking classes. So I'm not in the study that I showed, but other studies, and I've seen the sex difference in those studies as well, like in the meta-analysis, a lot of those studies would have used an active control. So those participants come into the lab, they go into the gym, and they actually work as a control group with other people, and they do balance exercise, balance and tone exercises. And so that's an active control. They also get the social interaction, and that's why we have that type of control group. But even in the randomized control trial that I did show that had the sex difference, that control group was an education control group, but they still came into the lab and they in a big group, and they did really fun classes. Actually, they did like cooking classes together with a nutritionist, and they did like cognitive, like they did a lot of different things in a group setting to sort of get at that social interaction part.
It's so interesting, actually, to think about that, Cheryl. Go ahead.
No, I think that's great. I'm just wondering, I'm just thinking of the way I walk sometimes with people, and, the people that I choose to walk with, I don't, I'm not part of those big running groups or anything, which is good, and you do get social interaction, but the people that I tend to walk with, are people that I'm close to, and I'm working out different problems in my life, and I'm problem solving as I'm walking, and it's a different I guess mental status of, how, and I'm just wondering if that could actually play into dementia, because if you're solving problems, then you're not getting stuck in ruts. I don't know. It's
absolutely.
Almost like little therapy sessions.
Yeah, no, I absolutely agree. And there is like, a PhD student finished last year, and she did a cognitive, so sort of similar, what you're sort of in the idea of like talking and problem solving and just using your brain more, because while you're doing the exercises, if that's sort of what you're getting at. She did a study where it was a randomized control trial, and she did cognitive training. So, but this is more like specific cognitive training. So they're actually doing problem solving on the computer and playing these games that use their brain. But then she paired it with exercise and without exercise to see if that exercise bit could actually make that even more so, like bolster that effect of the cognitive training, or like just using your brain a bit more. And then other things that we can actually look more at social interactions in some of these studies, because we do collect social interaction questionnaires and see if that might differ even between the two groups or might moderate the relationship. It's definitely an excellent thing to look at.
Well, the other thing I was thinking about, Cheryl, that's why I was interjecting, because when I think I walk the same way Cheryl does. So I walk and rant with my neighbor, and we both rant, and then we solve our problems. But I'm wondering if it also might have stress might have a role there. Right? Cortisol, stress, right? Mm-hmm. If you're walking, and even though I know exercise can increase cortisol levels, if you're very stressed, then you go for a walk to try to, and then he said this, and then my husband did this. Can you believe it? by the end of the talk, perhaps, talk and walk, right? So I'm wondering how that might play a role. Do you know? Do you have any?
And I'm wondering, if it's kind of the differences between the way men walk and the way women walk. Like, I think women, I don't know. I'm not a man, so I don't know if men walk the same way that I walk, if they walk with a friend and they rant, and like you say, it's like they're kind of problem solving, or if they go off on their own and they listen to music and they solve on their own, and when they're together and they're walking, they're not talking about intimate details of their lives. I don't know.
Yeah, I mean, I never, even in the randomized control trial where we did moderate paced walking with them, anecdotally, I would say that there's definitely chit-chatting going on, even though it's hard because it's moderate paced, and we make sure they are walking at this very, fairly intensive. Moderate paced for an older adult is pretty, it's intensive enough. And so there is definitely chit-chatting going on. Whether it affects cortisol levels, I think that would be very intriguing. We can maybe potentially look at that in one of the studies that we're doing right now, because there is aerobic training in that study, and I'm collecting saliva to look at cortisol before and after one of their classes. I do that at the beginning, like before they start exercising the training, and then they do it in the middle and at the end. And so, at some point I can look at to see if cortisol is different if they are doing the aerobic training, because they all do it in the group setting anyways, and they're walking and they are social interacting with each other, and the control groups are doing other stuff, but they're not doing the exercising, and they're not getting the same experience. So I think we can maybe start to look at that, but it's an interesting idea of the type of cortisol, not the type of cortisol, like we know exercising increases cortisol, but sometimes it doesn't seem to have a bad effect, but cortisol, we always think of having a bad effect.
Chronic stress versus
chronic
acute
stress.
and all
Yeah.
that.
So there's a lot of stuff to look at there.
We have a couple more questions in the chat that I wanted to get to. Melanie Butt is asking, who are the authors of the study you just mentioned with the cognitive training versus exercise versus both? I know there's somebody in McMaster.
Oh, who's the
Yeah,
first
the author.
author
Is it
of that one?
published, or is it?
Yes, it's published. So there's a couple of papers that have come out of it. It's Fit Brains is what they use. That's Lizana Tenbrinky. I'm saying her name totally wrong because I'm trying to say it the way it's spelled so you can write it down. But you can also look Teresa Lou Ambrose. So it's that's our supervisor. So it's out of her lab actually.
Put it in the chat maybe.
Oh, sure.
And then I'll ask this question slowly while you're typing it in the chat. Lauren's asking or saying that if you have any help, if you want help with court, you can ask, which is fantastic, Lauren. We're going to come back to you. Faith is asking, you mentioned estradiol increases BDNF. Have you come across any studies, female or male, with hormone receptor blockers or disruptors, and seeing whether that modulates any effects on exercise efficacy?
I'm just going to read it here. Do you have come across studies of female or male hormone disruptors? So the only one I can think of off the top of my head is an older study done by Cotman that looked at overrectomized versus non-overrectomized rats. But what's also interesting, that just popped in my head, is the idea that at least in mice, males and females actually run at different levels. So females run a lot more depending on their estrogen levels. So when estradiol is high, they run way more. So it's kind of interesting. But I don't know of any that looked at hormone disruptors.
Well, I think the overoptimized studies, that's a good one to, yeah. But you didn't, so what happened in the OVEX group, I think BDNF, well, I know what happened, but I've just finished that thought with the Cotman paper.
The Cotman paper, the overoptimized mice looked more like the male mice, and they also exercised less, but they didn't look at BDNF levels. So I don't know if BDNF had anything to do with that. I mean, I suspect, yeah, that's why the exercise wasn't as effective in the overoptimized mice.
I swear they did, but maybe I'm thinking of another study now.
It's an older one, and I can't even remember the year. No, it's called
Cotman or something, I think. Yeah. Yeah, I know that's true, Lauren. There's a lot of studies looking at aromatase inhibitors in breast cancer, like letrozole, and whether there's something there specifically on efficacy. I don't know. I don't know that people, especially like exercise efficacy on cognitive function from following those. It's a good. I mean, the data should be there, so that would be kind of
we could look at it if they even within the physical therapy department here at UBC, working with Kristen Campbell, and I think she does take blood, but how we look at depends on when we took the blood and stuff. So definitely we could address that question, I think, in a way.
Yeah, that's really interesting. Well, now you have, you better get that faculty job so that you can do these 20 years of studies that.
Yeah, Cheryl, I'm going to look at your social interaction idea. I'm going to look at the cortisol idea and the aromatase inhibitor.
I have a quick question maybe, and then I'll let everyone go. Well, you guys can go whenever you want, but global cognition versus executive functions. Why? Like what does that tell us in terms of, because I think the males and I can't remember which one it was, the ABC study or which one it was, but for males it was, and females there was a benefit to global cognition, but for executive functions it was just in females. I think the walking, is that true?
Yes. So we see that, I've seen that in several different ways now that it seems to be specific to executive functioning, and not that the sex differences. And so why is it executive functioning? I mean, part of it could be the global cognitive metric. The test is not great. It's just like it's not a very good test for anything specific. It's I think in that one was the MMSE, so not a great metric. But I do think there's something there in terms of executive functioning versus even hippocampal dependent memory tests. I'm not seeing the sex difference in exercise effects on even memory tests. I'm seeing it just for executive functioning tests. So why? I don't know. I don't know yet. I mean, I think that's the next step, is to figure out why it's executive functioning. I mean, I think it probably has something to do potentially with differences in connectivity between in males and female brains, that are in the connectivity in the areas that are subserving executive functioning. Could have something to do with the level of BDNF receptors in those different areas between males and females. It could do with, I mean, that there's just a lot of different hypotheses. I don't actually have an answer to that one yet.
I was just wondering too about your cardiovascular stuff. Maybe there's something to do with, I don't know, vascular innervations in either areas and benefiting, one versus the other sex. Anyway,
yeah, I don't know.
Super, super interesting. So with that, I know we're 4 minutes early, but then I can give you back 4 minutes in your day. Although I have more questions, but I can ask Cindy anytime. But thank you so much, unless there's another question that comes up. Cindy, thank you so much for your presentation. It was really beautiful slides and really clear. So thank you so much. And if everyone would join me in giving Dr. Barha another round of applause. We've got a lot of other things in the chat there too.
And if anyone has anything else they want to talk about, you can always get me on email. I put my Twitter up there, which I'm trying to use.
Thanks, Cindy.